You have to wonder, why is quitting alcohol so incredibly hard for so many people? It turns out the answer isn't just about willpower, it's actually written into the chemistry of our brains. Today, we're basically going to be scientific detectives following the clues that are uncovering a major player in the suffering of alcohol withdrawal. And what's really cool is that it might just point us toward a whole new way to treat it.
So to really get why this research is so important, you first have to understand the sheer scale of the problem. We're talking about alcohol use disorder, or AUD. And in the United States alone, well, look at that number. Over 29 million people are affected. That is a huge number of people facing this challenge, which is exactly why finding better ways to help is so absolutely critical.
And for so many of those 29 million, maybe the single biggest hurdle they have to clear is the brutal experience of withdrawal. I mean, it is so punishing, both physically and emotionally, that it just creates this awful, vicious cycle. It often drives a person right back to drinking just to make all that pain stop.
And let's be clear, we are not talking about a bad hangover here. Withdrawal is a whole different beast. It brings on this storm of really severe symptoms. You get things like hyperalgia, which means your sensitivity to pain is cranked way up. Even little things hurt a lot. On top of that, you have this crushing anxiety and just a deep, constant feeling of negativity that is so hard to live with. So that brings us right to the central mystery, right? The big question that scientists are trying to answer. We can see all these awful symptoms on the outside, but what is really going on deep inside the brain? What's changing in there to create all this suffering? Because if we can answer that, we might just find a better solution. To crack this case, researchers needed to narrow things down. And over time, they've really zeroed in on a prime suspect, a particular brain chemical that seems to be right at the scene of the crime. It's called orexin. So what is orexin? Well, a simple way to think about it is like it's the brain's main go signal, or maybe like a master amplifier. It's a type of chemical called a neuropeptide, and its job is to dial up a bunch of really important functions, like how alert we are, our motivation, and especially how we respond to stress. It basically tells the brain, hey, pay attention, it's time to act.
Under normal circumstances, Orexin is kind of a jack of all trades. It helps manage our sleep-wake cycle, controls our stress response, pushes us to seek out rewards, and keeps us motivated. But you can probably already see how a system that has its hands in all of these pots could really go haywire during something like addiction and withdrawal.
And here it is. This is the first big piece of evidence that connects our suspect, Orexin, to the crime. In studies with people who have alcohol use disorder, researchers found that when they go into withdrawal, their orexin levels shoot up. And not just that, the higher their orexin levels, the worse they said their anxiety and cravings were. That right there is a massive clue.
Okay, so we've got a clue, a really strong one. But a correlation, even in humans, isn't quite proof of cause and effect. So to really build their case, scientists took the whole investigation into the lab. They wanted to see if they could connect Orexin directly to withdrawal symptoms in a really controlled way.
So here's how they did it. It was a pretty straightforward, methodical plan. First, they made female rats dependent on alcohol by exposing them to its vapor over a long time. Then, they cut off the alcohol, which threw the rats into withdrawal. Next, they tested them for behaviors related to pain and anxiety. And for the final step, they looked directly at their brain tissue, the scene of the crime, to search for biological clues.
Now, the scientists were also pretty clever about the setup. They actually used two different age groups, adolescent rats and adult rats. And that's really important because we know alcohol can affect the brain differently depending on your stage of life. So for the younger adolescent rats, they focused on testing for pain sensitivity. And for the adults, they looked for anxiety-like behaviors. And they didn't just look at the whole brain at random. Nope. They focused their search on a very specific, very important neighborhood called the ventral tegmental area, or VTA. You could think of the VTA as like a master control center for reward, motivation, and addiction in the brain. If Orexin was stirring up trouble, this was a very likely place to find the evidence.
All right, the stage is set, the experiments have been run. So now it's the moment of truth. What did the scientists actually find when they put all the pieces together, from the behavior to the brain tissue?
So the first big piece of evidence came from the young rats. What they saw was that during withdrawal, these adolescent rats were way more sensitive to pain. When exposed to a little bit of heat, they pulled their paws away much faster than the rats who weren't in withdrawal. That's a textbook sign of that hyperalgesia we were talking about.
And the evidence from the adult rats?
it was just as striking. They put them in a special maze, and the rats going through withdrawals spent way less time exploring the open, exposed parts of it. They preferred to just hunker down in the safe, enclosed areas. And in the world of rodent research, that is a classic indicator of anxiety.
Okay, so the symptoms check out. The lab animals are showing the same kind of pain and anxiety that we see in humans. But that just brings us right back to the million-dollar question. What's the biological fingerprint here? What is physically changing inside the VTA that's causing all of this?
And this is where they found the biological smoking gun. When they looked at the brain tissue, the rats in withdrawal had a significantly higher number of receptors for orexin in their VTA. Think of it like this. Their brains had literally built more landing pads for that orexin go signal, making that part of the brain extra sensitive to it.
And it wasn't just a random increase either. The scientists found a direct link. It was almost like a perfect line on a graph. The more orexin receptors an animal had in its VTA, the more intense its pain sensitivity was, or the more anxious it acted. That is a really powerful connection between the biology and the behavior.
But to really truly close the case, they had one last question to answer. They had a correlation, a strong one, but could they prove causation? In other words, is just flipping the orexin switch in the VTA enough, all by itself, to cause these negative feelings, even if there's no alcohol involved at all? So they designed this last test, and it was just beautifully simple. They took perfectly healthy rats that had never, ever been exposed to alcohol. Then they injected a tiny amount of orexin right into their VTA to basically turn that system on, and then they just watched to see what happened next.
And the result? It was completely conclusive. Just by activating those orexin receptors in the VTA, these perfectly healthy, alcohol-free rats immediately started acting anxious. And that confirms it. This specific brain circuit doesn't just contribute to withdrawal, it can actually generate these negative feelings all on its own.
So after following all the evidence, from the human studies all the way down to the individual brain cells in the lab, we finally reached a verdict. And this verdict points towards a really exciting, really promising new path for helping people.
So here's the bottom line, the main takeaway from all of this. The orexin system, in this one specific part of the brain, the VTA, it isn't just related to withdrawal symptoms. This research shows it's a key, fundamental, biological driver of the pain and the anxiety that makes quitting alcohol so incredibly difficult.
And that leaves us with one final and I think really hopeful question. If an overactive orexin system is the problem, what if we could just find a way to temporarily turn it down? Could a future treatment, a medicine that specifically blocks these orexin receptors, actually relieve the suffering of withdrawal and make it easier for people to break that vicious cycle for good? Based on this research, that is a very real and a very exciting possibility.